leaves me to introduce our opening speaker, Professor Ammar Al-Chalabi. And he does not need any introduction speech, but Ammar is an NIH Senior Investigator, Professor of Neurology and Complex Disease Genetics at King's College London. He is the co-director of the UK MND Research Institute, the King's MND Centre Director, and he chairs the International Symposium of ALS-MND. He's also co-leads the Project MIND International Whole Genome Sequencing Consortium and the MND Register of England, Wales, and Northern Ireland. And I have to also say that Ammar's work, as we all recognise, have had multiple honours, including the Forbes Norris Award from the International Alliance of ALS-MND Associations, the Healy Centre and International Prize for Innovation in ALS, the Sheila Essay Award from AAN, and the Fellowship of Academy of Medical Sciences. So we're all extremely, extremely thankful to have you here, and we're really looking forward to your insights on what causes MND. So I'd like to invite Ammar Al-Chalabi to take the stage.
Thank you very much, Tanuja. So thank you very much, Tanuja, and thank you to the organisers for inviting me. So I'm going to be talking about what causes motor neuron disease. And throughout my talk, I will be using ALS and MND interchangeably. So we know that there are two obvious causes of ALS. One is, excuse me, I'm just going to turn this. One is family history, and the other is age. And we know that a family history of ALS is present in about 5 to 10% of people, depending on how you define a family history. And that tells us that there is very likely to be a genetic contribution. And we know that the risk increases with age. So the older somebody is, the more likely they are to develop motor neuron disease. And that therefore tells us that whatever the mechanism is, it must be something to do with age in some way. Not necessarily directly, but aging must be involved in some way. So let's just look at the family history aspect. This is a pedigree from my colleague, Peter Anderson. The actual true pedigree is about 400 people, and it's about 20 meters long when printed out on normal paper. This is a small selection of it, and it's been circularized. So if you're not used to looking at pedigrees, the way they work is the center in this case is the first generation, and then every ring outwards is the next generation. Any person that's shaded, so anyone that appears in black on here, is affected. And we've only shaded the people with motor neuron disease. This family has a single disease gene as the responsible gene in for their motor neuron disease. That gene also causes frontotemporal dementia, but as I said, I'm only showing ALS. Now, the group highlighted in red there, you can see that there's somebody affected, then it misses the next generation, and then there's a father-son transmission as well. So if you saw the youngest person in that affected in your clinic, you would think this person has familial motor neuron disease. Their parent is affected, they've got a great-grandparent affected. So that looks like what we would call familial ALS. And if you look at this lower part of the pedigree, those are third cousins who are affected. And most people don't know who their third cousins are. And if you saw one of those people, you'd think, well, they don't have a family history of motor neuron disease. But that's because this particular gene hasn't expressed itself in the other generations. We call that reduced gene penetrance. So the penetrance of a gene is the probability of developing whatever that gene causes if you carry it. So this is really to show you that ALS is complex genetically. Carrying a disease gene isn't enough to cause the condition, but it is enough to push us towards getting that condition. If you do genetic testing in people with no family history, and here I'm using the word sporadic to mean people with no apparent family history, it doesn't mean it's not genetic, it just means there's no family history. If you test people at different ages, then this is the probability of finding an clinically actionable genetic result at any age. So you can see that in general, it's about 20%. So if you test people with no family history, about 20% of them will have a genetic test result that would require some kind of clinical response. And you can see that's true even up to people who are quite old. Now, usually we're taught, at least we intuit, that genetic factors are only there when you're younger, that it's more likely if you're younger. But you can see here that there's a genetic contribution really regardless of your age. So knowing that, I think 20% actionable genetic results is quite a high frequency. That means that we should really offer genetic testing to everybody who comes to clinic, with appropriate counseling, of course, but it should be offered to everybody. Now, if we look at genetic ALS, we do know that it is more likely to present at a younger age. In fact, in our population, if we look at people with a family history compared to people without a family history, it's about a 5 or 6 year difference in age. But is that just because people with a family history know what they're looking for? And so if someone's affected, they'll go to the doctor earlier. That's the most likely explanation. But you can address that by looking at the people who have no family history. Do they carry a genetic variant or not? Now, if you look at that group of people, so the people with a family history and the people who have no family history but do have a genetic cause, then in those people, there's no difference in the age of onset. They both have a young age of onset. And if you compare the people who have no family history but a genetic cause with the people who have no family history and no obvious gene, then there is a difference in their age of onset, about 3 or 4 years. So that tells us that it's not an ascertainment bias. I mean, there might be a little bit of ascertainment bias, but the real cause of the younger age of onset is actually that there is a strong genetic contribution, that somehow carrying disease genes lowers your age of onset. The other important thing to consider is that generally we look at causes of ALS. In fact, that's the title of this talk. But should we really care about what causes ALS? Because really, when we see people, they're coming to us after they've been diagnosed. The cause has already happened. What we really care about is prognosis. Is there something we can do to treat ALS once it's started? And that, you might think they're the same thing, but they're not necessarily. And an easy way to consider this is, imagine if you have a bonfire and you pour lots of petrol on it, and then you light a match and you ignite the bonfire. If you blow out the match, which is the cause of the bonfire blazing, it's not going to make any difference to whether the bonfire is blazing or not. It's still going to be going. So something that causes and something that progresses are not necessarily the same thing. They might be. Obviously, if you blow out the bonfire with enough of a blast, then that would have an effect. So one thing we did was to look at whether there is a disconnect in genes between risk and survival. That matters because then you can target your drugs at the genes that are causing the progression rather than the risk. The gene we looked at is SOD1. Now, SOD1 gene variants are found in about 2% of people with motor neurone disease. And what you can see here on the x-axis is the different positions in the SOD1 gene where you can find a variant, and they're named, the number is the codon at which that occurs, so the amino acid where that's happened. And then the first letter is the original correct letter, the common letter that's seen in the general population, and the second is the one it's been changed to in the amino acid code. The two horizontal lines represent survival. So the y-axis is showing survival, but it's logarithmic. So the first line, the lowest line, is at 2.5 years. So everyone below that line has lived less than 2.5 years. The second line is the 5-year survival. So everyone above that line has lived more than 5 years. And what you can see is that some of these gene variants are consistently associated with aggressive disease. So if you look at the A5V variant, which is the most left one there, that's consistently associated with very aggressive disease. Everybody, nearly everybody, has survival less than 2.5 years. But then if you look at the D91A, which is the middle one, that variant is fairly consistently associated with much less aggressive disease. People generally live more than 5 years. And then if you look at I114T, which is the rightmost one, it's got a complete spread. So even within the same gene, you don't necessarily have consistent relationship. All of these will increase your risk of ALS massively, but they don't necessarily have the same effect on your survival. That's even within the same gene. So The genes we've been talking about so far are what we call large effect genes. They have, if you carry that gene, it greatly increases your risk of ALS. It doesn't just nudge you towards getting it. It really means you've got a very high chance. And those are what we call Mendelian genes. They've been catalogued in various places. So the ALS Online database, for example. There's also a question about whether something that's published is always an ALS gene. There's some contradiction in there, you need replication, etc. Not everybody will believe every gene is an ALS gene. So there's a panel called the ClinGen Adjudication Panel, which is an international panel that meets roughly once a month and goes through with a proper systematic process the evidence for each gene so that they can decide whether or not this has strong evidence, limited evidence, etc. And then that's made publicly available. And that's continuously updated. And then the same kind of question about ALS variants themselves. Within SOD1, for example, most people regard any variant as an ALS-causing variant, but we don't necessarily know that. And as we come to other genes, you get variants of uncertain significance, where you have a change, but we don't know whether that really is important for that person in terms of having caused their motor neuron disease. There may not be enough evidence. Just because you have a variant in that gene, and it's an ALS gene, it doesn't mean that variant necessarily was responsible. It may make no difference to that gene. So you have a variant curation expert panel in ClinGen as well, that again goes through systematically the evidence for every variant. So you can imagine this is a lot of very tedious work, but there's a huge international effort to go through that. What you can also see here is a graph on the right of the gene discoveries. This is just until 2017. And you can see that the rate of gene discovery is roughly doubling every 4 years. Now, on this graph, the big spheres, the size of the sphere is proportional to the number of publications for that particular variant. So it gives you some idea of the importance. So the big spheres are important genes, and the smaller spheres are less important in some way. And red represents people with no family history, and blue represents people with a family history. So you can see essentially all of those variants are found in people with and without a family history. And the other thing you can see is the names are color-coded, and the color coding represents different aspects of what pathway that gene might be involved in. So do all of those genes end up with a common mechanism? We know the final common pathway is motor neuron death, but is there a common mechanism that leads to that motor neuron death? And one of the things which I'm sure you'll be hearing about throughout this symposium is that about 97% of people with motor neuron disease have a protein that's mislocalized in the cell. And that protein is called TDP43. It's coded by a gene called TARD-BP. And TDP43, when it's in the wrong place, it has a number of effects on the cell, but one of the most important appears to be that splicing goes wrong. Now, splicing is where genetic information is converted into essentially a photocopy of the gene, we call it RNA, and then that is stitched together to make the instructions for a protein. And if that goes wrong, then there's a splicing error. So TDP43 is very important in making sure that splicing works correctly throughout your DNA for nearly all genes. But one of the things that's more recently been discovered is that lots of genes have hidden sequences that aren't normally read. They're normally switched off. It's a bit like if you have a recipe for a cake, for example, and in the middle of that recipe, say it's a chocolate cake, and in the middle of the recipe is a section on adding beef into the chocolate cake. It's crossed out or it's masked in some way. If TDP43 isn't in the cytoplasm, then when you read the recipe, you'll start adding beef to the chocolate cake and it won't taste quite how you're expecting. So those are called cryptic exons. And if TDP43 is in the wrong place, then those cryptic exons are unmasked. One of the important gene risk factors that we know exists for motor neuron disease is called UNC13A. And changes in the UNC13A, there's a particular region in it called a SNP. And if you carry two variants of those SNPs, one from your mother and one from your father, then you have an increased risk of motor neuron disease, only a little bit, but it does increase your risk, and you tend to have a faster disease progression. And it looks like TDP43 normally will bind particularly to that site in the gene. So if you've got a variant there, it affects how it's going to bind. And if you don't have TDP43 in the right place, and you have those variants, it makes even the little bits of TDP43 that are in the right place not work so effectively. So it's an example of where we're beginning to unravel how all these different bits of the puzzle fit together. So we've talked so far about these large effect Mendelian disease genes, but are there other genetic factors that will increase your risk? So one way you can look at that is to look at what's called the heritability. So heritability is, it's not the inheritance, it's the tendency, it's the variation in genetics that accounts for the variation in whether you have something or not. And one way you can address that is by looking at twins. So identical twins share all of their DNA and essentially all of their environment, at least for the early years of their life. Whereas non-identical twins share about 50% of their DNA because they're siblings. And again, you can argue they share their environment, at least in the first years of their life. And so the main difference between identical and non-identical twins is genetic. And that means if you look at the frequency of a condition in identical twins, how commonly do they, are they concordant? Do they show the same outcome versus non-identical twins? It gives you an indication of how much genetic variation is contributing to that clinical picture. So if you do that in motor neurone disease, then you can see that in identical twins, for this particular population, about 5 of them both had motor neurone disease. Whereas for the non-identical twins, that was never true in this particular population. And where they were discordant, where only one had motor neurone disease, 44 of the identical twins, that was the case, and 122 of the non-identical twins, all of them, that was the case. And if you do the maths, that tells you that the genetic variation contributing to the risk of motor neurone disease is about 60%. So that's additive genetic variants. These are small genetic variants that each of them isn't enough to cause motor neurone disease on its own, but each of them pushes you towards it. Maybe they increase your risk by 1% or 2%, that kind of thing. But if you've got 20 or 30 of them or 50 of them, that's a significant boost to your risk of getting motor neurone disease. The way you can detect these is by looking at association studies. And to do association studies, because you're looking for small effects, you have to look at huge numbers of people. And also, because you don't know where these variants are, you don't have any clue, you have to look at hundreds of thousands or millions of genetic variants in each person. When we do that, so this is an example where we've looked at 27,000 people with ALS, with motor neurone disease, and nearly 111,000 people without. And we've tested them for 10.5 million genetic variants. On the graph, every dot represents an association test. And the higher up on the graph that test is, the more significant it is, the less likely it is to have been seen by chance that there was a difference between the cases and the controls. And the ones in red, the peaks in red, are the ones that we think are important, the ones that pass all of our tests. You can see the skyscraper in the middle is for a gene called C9orf72. And that gene is actually responsible for that very first family I showed you, where not everybody develops motor neurone disease, but it gives you about a 40% contribution to your risk. So it's quite a high risk. The penetrance is about 40%. But you can see that there are lots of other variants there. So the ones in red, those are Mendelian genes. So we can pick them up with these association studies, even though they're designed to detect these small effects. And the ones in green are newer variants that have only been found recently. Before that, we knew already the black ones. You can see UNC13A there, which is that gene I was telling you about before with the cryptic exons. If you look at, in general, these association studies, you can come up with some pathways where the genetic variants tend to occur. And they tend to occur in pathways involved in cholesterol metabolism and in the immune system. They occur in others as well, but those two pathways I think are quite interesting. You can also look at other types of genetic variants. So in the genome, you get little sections that are repeated. All the variants I've shown you so far are single point variants. So you just have one letter that's switched in the DNA code. Here you've got sections of letters. So for example, it may be CA. So instead of having 3 or 4 CAs like most of the population, maybe you have 40 CAs. So go CA, CA, CA 40 times. Those repeat sequences also increase the risk of neurological disease. Quite often they're triplet repeats. Quite often they're CAG. They don't have to be. They can be any number of repeats, but often they're triplets. So ataxin 2 Normally, if you have a very large number of repeats in ataxin-2, then you have a risk of a condition called spinocerebellar ataxia. And that's a condition where you become uncoordinated because the cerebellum, which is the back part of the brain, doesn't work so well. If you have expansion in C9orf72, which was that big signal I showed you in that slide just now, and also responsible for that first family, that greatly increases your risk of motor neuron disease. You can also have repeats in NIPA1 and in ataxin-1. So repeats seem to be important for some people. The graph, the table you can see on the right, is looking at ataxin-2 repeats in motor neuron disease. Now, I told you already, if you have a large number of repeats, then you get spinocerebellar ataxia. If you have a normal number of repeats, then you don't have anything. But if you have something in between, an intermediate size repeat, that increases your risk of motor neuron disease. And when this was first published, there was a lot of skepticism. People said, this is a crazy idea. Why are you saying that if you have a normal range, you're fine? Then if you have something in between, you get motor neuron disease, and larger gives you spinocerebellar ataxia. There was a lot of skepticism in the community. But every single line here, in that graph, is a different study looking at ataxin-2. And you can see they all tend to be on the right-hand side of that middle line. They're all showing a positive association. And in fact, on average, it roughly triples your risk of motor neuron disease, having an intermediate size repeat. You may not think that's a lot, and you could argue it isn't. But it means that you've got about a 1% chance of developing motor neuron disease if you carry an intermediate repeat size. You also have another type of variation, which is called a structural variant. And that's where large sections of the genome are copied. So you may have whole genes copied, or they may be twisted round, so they're backwards, or you may have a big block of something inserted into it or deleted. And you can get an insertion in a gene called ERB-B4, which is quite common in the general population. So about 25% of the general population has this insertion. If you happen to have motor neuron disease, about 1% of people have respiratory onset. So the first symptoms they get are with breathing. And about 70% of those people have this insertion. So that suggests that it may be nudging people towards having a different presentation of their motor neuron disease, which is another complication when we're looking at how genes increase your risk. The other thing that seems to be slightly unusual is if you look at telomere length. Now, telomeres are caps on the end of your chromosomes. Remember, chromosomes are what your DNA is carried in. When you duplicate your chromosomes, if they didn't have these caps on the end, then the last bit wouldn't be copied properly. So you have a repeated sequence on the end of your chromosomes, very, very many hundreds of times. And that means that your cell copying machinery can just read through that, and you get a kind of cap on the end of your chromosomes. And the length of those telomeres changes as we get older. They shorten as you get older. And people with shorter telomeres are more at risk of cancer. But for some reason, people with shorter telomeres have a reduced ALS risk, even though they shorten with age, and ALS is an age-related disease. And they have a slower condition, a slower rate of progression. And there's also an interaction if you have C9orf72 as the cause of your ALS. So there's a kind of additive effect if you've got both. So just to summarize, genes are important in ALS regardless of your family history. The large effect Mendelian genes are found in about 20% of people, regardless of family history. The small effect common variants give you about a 60% contribution to your risk. And if you add all that together, there's more genetic ALS in sporadic ALS than there is in familial ALS. Because if you do the numbers, about 8% of all ALS is familial genetic, compared to about 18% of the genetic contribution from sporadic. So now let's look at age. The risk of ALS increases with age. And that tells us something about mechanism, as I said. So this is a graph of your risk of ALS at any age. And you can see that men always have a higher risk than women. And that on average, by the time you're about 85, there's roughly a 1 in 300 risk. So it's quite high, it's 0.3%. That's different from the incidence, because remember, the incidence is the number of people you would see age adjusted per 100,000 person year. So it's a strange unit. This is a more intuitive unit, just what's your risk at any age. So if you live to 85, you've got about a 1 in 300 chance. So roughly 1 in 300 people will die of ALS. So we can make a kind of model now. We can say that everyone is born with a genetic load that predisposes them to ALS, and that with time, you get some kind of cell damage, and then there may be some environmental risk factors. We haven't really spoken about those yet, or maybe some random factors we don't know. And unless you reach a particular threshold of burden, you won't get ALS. But if you do, then at some point, if you hit that, you'll develop this self-perpetuating process of ALS. So that's kind of gene time environment model of ALS. So what are the possible environmental risk factors, and why haven't we found any? It's very difficult to study environmental risk factors in ALS. Looking for genetic factors is a huge task. There's 3 billion letters in your genome, and you've got to look in thousands and thousands of people to find a result, but it's a finite problem. If you're looking for environmental risk factors, there's no easy way to do it except for asking people about what happened. And people's memories vary. And there are almost an infinite number of possible environmental questions you could ask. Could it be that you eat apple cores, or that you sleep with your neck in a funny position, or that your shower's too hot, or there's anything could be. And so that makes an extremely difficult, challenging issue. So we look at common types of things that we know cause other diseases like smoking, exercise, heavy metals, pesticide, military service. Those are things that have been looked at in ALS, and many studies have suggested they increase your risk, but not all of them. It's inconsistent. And there's variable evidence, and it's difficult to replicate them. So we've got a number of ALS mysteries. Genes that cause ALS also cause other diseases. I've already told you that the C9orf72 expansion mutation, for example, causes frontotemporal dementia as well as ALS. Not all gene carriers develop ALS. So you've heard about this thing called gene penetrance. You can carry a gene, it doesn't give you ALS, it just nudges you towards getting it. Environmental causes are difficult to find. We've looked, but we can't replicate all of them, and there's no definite environmental risk. There's an increasing risk with age. There's no obvious single cause of motor neuron disease, and there's this age-dependent penetrance, as I said. Those are all properties that are also shared by cancer. So can we use a cancer model to ask whether ALS is similar in some way? Now, in cancer, there's a thing called the multi-step model, where you have to have multiple hits before you develop your cancer. And if there is a multi-step process, then it follows a power relationship between the age of onset and the incidence. And that means if you plot the log of the age of onset and the log of the incidence, you'll get a straight line. The slope of that line will give you information about the number of steps involved. So we looked at this in the Southeast ALS register, which was a register covering about 3.5 million people in the UK. And we saw what looks fairly close to a straight line. The R-squared value is 0.95, so that's a good indicator. Once it reaches 1, then it's a perfect straight line. And then we contacted our colleagues in the Netherlands who had a population register, and in theirs, it's 0.99, it's much closer to a straight line. Our colleagues in Italy, 0.95, in Ireland, 0.99, and in Scotland, 0.97. And then if you put them all together, it's 0.99, so very close to a straight line. But the other thing is, because we put them all together, that means they must have all had the same slope because it's the same line. And if you look, the slope is around 5. So the slope is 1 less than the number of steps if you do the maths. So that tells us that it must be a 6-step process. You need 6 molecular steps for some reason to develop ALS. Doesn't mean it definitely is, but it's consistent with it. And that would answer a lot of these mysteries. ALS genes also cause other diseases. That's because, for example, maybe you need steps 1, 2, 3, 4, 5, 6 to get ALS, and 1, 2, 3, 4, 5, 7, and you get frontotemporal dementia, a different step at some point. Not all gene carriers develop ALS because you may not get all of the steps. Genes may give you some of the steps and then you don't have the others. Environmental causes are difficult to find, perhaps because different factors will cause the same molecular step. For example, smoking increases your risk of mutation, so does radiation exposure. maybe something like that. The increasing risk with age is because it takes time to accumulate all the steps. The fact there's no single cause is because different factors can trigger the same molecular step. And the age-dependent penetrance, again, because it takes time to accumulate all the steps. And this has been replicated a number of times, including here by Steve Vucic and Matthew Kiernan, and by Naomi Ray. So in the Australian register, it's been shown twice, in a Danish register, in a Japanese register, and twice in an Italian register. In South Korea, when this was done, it was shown to be a 5-step process. So that suggests it's slightly more aggressive. You need fewer steps. And Adriano Chio in Italy has a register with good genetic information. We were able to use that to ask whether genes account for some of these steps. And he could show that if you carry this variant in C9orf72, that means that you've got a 3-step model. So 3 of the steps are accounted for. Having that gene variant triggers 3 of the steps, and you only need 3 more steps before you get ALS. If you carry the TARD-BP variants, so those are the ones that code for TDP43, then that's a 4-step model. That means it's accounted for 2 of the steps. And if you have SOD1, that's a 2-step model. So SOD1 variants use up 4 of your molecular steps, and you only have 2 left before you develop the disease. So they really do strongly push you towards getting ALS. You can also look at it you can also use it to look at lifestyle. So for example, this is from Naomi Ray's work in Australia. So if you look at the black line, that's ALS in general, 6 steps, as we found with the other studies. If you look at ALS with heart disease, then it looks like a 7-step model. So it appears that heart disease protects you. And that's consistent with what we see in clinic. Lots of our patients don't have heart disease. But remember, heart disease increases in risk the older you get, and also is more common in men. So it could be that there's a competing causes of death thing. If heart disease takes people out of the population before they are old enough to get motor neuron disease, it would give you this kind of pattern. And so Naomi's group accounted for that very cleverly, actually, by looking at people with heart disease and in that population doing the analysis. And they showed that actually having heart disease uses up 3 of your steps. It's a bit like having a C9orf72 expansion mutation. So heart disease actually increases your risk of ALS. The reason we don't see people with heart disease in our clinics is because they're dying before they get to the clinic of their heart disease. So why is heart disease associated with ALS? Well, if you remember, I showed you before the cholesterol pathway seemed to be important. And you can use a technique called Mendelian randomization, which is using genetics to look for environmental risk factors in quite a clever way. And if you do that, then you can show that having high cholesterol increases your risk of ALS. And it seems to be causal. Having high cholesterol in some way causes your motor neuron disease. You can also use another genetic technique called epigenetics, where you look at methylation marks on the DNA. So the DNA is modified with methyl groups, and that switches on and switches off various groups, various genes in your DNA. You inherit some of those methylation marks, and others are affected by you and your environment. So it's a clue to environmental risk factors. And if you do that, again, cholesterol comes up in the pathway. You'll notice also inflammation, and that was the second thing I showed you earlier in our genome-wide association study. So we've got these 2 themes, cholesterol and inflammation, coming through. The other thing you can do is classify people with motor neuron disease, because we assume that even though we've got all these things causing motor neuron disease and they're all ending up in a similar final common pathway, we assume that it's one homogeneous condition because it always results in motor neuron death. But if it isn't, then that will affect which treatments we need to give. So one of the things you can look at is the brain transcriptome. The transcriptome is the amount of RNA you've got. It's the particular genes that have been switched on in that cell. And if you look at the way those genes are switched on and off in the brain, you can find that people with motor neuron disease end up in 3 different clusters. There's a cluster that seems to be important for synaptic and neuropeptide signaling. There's a cluster related to oxidative stress and apoptosis, and there's a cluster, again, related to inflammation. What we don't know is, are people always just in one cluster? If you start in the neuroinflammation cluster, does that mean you've got neuroinflammatory motor neuron disease and that's what you've always got? Or does it mean that you start with inflammation perhaps, and then at some point in the disease, it moves to a signaling problem, and then later on to apoptosis? Or maybe all three are relevant in everybody, but just neuroinflammation happens to be more important in you. Those questions are important if we're then going to treat. Because if you stay in neuroinflammation all the time, for example, maybe you need an inflammatory type of approach to your treatment. On the other hand, if you start off in one of them and then move to the other, then maybe we need to change the treatment we give at different stages. And if everybody's in all three, maybe we need a cocktail approach where we target all three mechanisms. What I like about this is you can also classify using the blood transcriptome. So this was done in brains, but if you take blood, you can actually get a pretty good classification. It's about 80% matching with what you'd find in brain. So neuroinflammation, again, has come up. So inflammation in ALS. We know that there is microglia and macrophage activation. So microglia are immune cells in the brain. And there's also a reduction in the number of T regulatory lymphocytes in the blood. Now, T regulatory lymphocytes are cells that are like the police of your immune system. They switch your immune system from being very aggressive to very calm. It's important that the balance is correct. If your immune system is too placid, then it won't respond to invading organisms. And if it's too aggressive, then it'll attack everything, including you. So you need to get that balance correct. And in people with motor neuron disease, the T regulatory lymphocytes are at low numbers and they don't work properly. So that's something to do with why the inflammatory process has taken place. Lower levels predict faster disease progression. We know that if you give interleukin-2, which is an immune switch, a protein in your blood that switches your immune system, high levels will switch off your Tregs and low levels will boost your T regulatory lymphocytes. So a trial has just finished in the UK using low-dose interleukin-2 as a possible treatment. We're waiting for the results of that, but the headline results were announced at a meeting actually about 2 years ago now, suggesting that taking into account how fast the disease progression was predicted to go using neurofilament, that low-dose interleukin-2 was a treatment that could slow down motor neuron disease. So this might be an important approach, tackling the immune system. So that suggests then that we've got multiple steps now. We've got an immune genotype, inflammatory triggers, and inflammatory response. So maybe it fits with the model. Retroviruses. 59% of people with motor neuron disease have a retrovirus signature in their blood in the form of reverse transcriptase. Only 5% of the general population does. Now, retroviruses, you'll all be familiar with HIV. That's an infective retrovirus. But actually, what you may be less familiar with is the fact that 8% of your genome is retroviruses. That contrasts with just 1% that codes for your protein. So 8 times as much as codes for protein codes for these viruses that have just integrated into our genome through evolution over millions of years. And they can be activated. Now, when they're activated, it's not the full virus that's activated, it's little bits, because the viruses have been damaged through evolution. So they don't make a proper functioning virus, they just make bits of it. So it's a bit like having a vaccination. But the bits that can be active, we do know can trigger a motor neuron disease-like condition. They damage cells. What we found was 5% of the general population that has these circulating things, these circulating retrovirus signals. If you look at relatives, you'd expect them, because they don't have motor neuron disease, to also be in that 5%, but they're not. About 50 to 60% of relatives also have activated retrovirus. And spouses, on the other hand, do match the general population. So that tells you that the retrovirus signal we were seeing is an inherited retrovirus. It's not an infective retrovirus. So motor neuron disease isn't infective, but these retrovirus sequences that we all carry in our DNA do somehow matter. One of our colleagues in the US, Avindra Nath, has done a lot of work to show that one of the most recent retroviruses, HERV-K, HERV is human endogenous retrovirus, that may be a cause of motor neuron disease. He's shown that if you put the envelope sequence of HERV-K, which is the bit that tends to be expressed in humans when we express it, that's toxic specifically to motor neurons. If you put it in cells, human cells cultured in a dish, they don't like HERV-K. If you put it in mice, then the motor neurons specifically are attacked. So we've got a clinical trial. Actually, it's also been going in Australia. It's literally just ended this week for recruitment using Triamec, which is an antiretroviral drug, to see if switching off the ability of these viruses to do anything in the genome might be a way of treating motor neurone disease. But I want you to look at this, which I think is very interesting. It's a mouse. It's a naturally occurring mouse. So it's not a mouse model that you make in a lab. It's a naturally occurring mouse that gets motor neurone disease. Now, this mice have endogenous retroviruses as well. They're called MERVs, murine endogenous retrovirus. And mice have different numbers of copies, just like humans do. And so you can get mice with very high copy numbers of endogenous retrovirus. And mice have genetic switches, just like humans do. And those switches can change how well those virus sequences are expressed. So some mice have a genetic variant that means not many of those virus sequences are expressed, and others have a switch that makes lots of those viruses be expressed. Now, if you happen to be a mouse that has multiple copies of endogenous retrovirus, and you happen to inherit one of these gene variants that means you express a lot of it, and you are then infected with murine leukemia virus, then you develop motor neurone disease. So this is a naturally occurring mouse model of motor neurone disease that is combining genetics, because they have this genetic switch and the copies of the endogenous retrovirus, environmental factors, they get infected by murine leukemia virus, and they have the endogenous retroviruses, so it explains the signal we see, and it's a multi-step model. So it's quite an interesting way of thinking about motor neurone disease. So let's see if we can stick all of this together now. So this is the gene time environment model I showed you earlier. Now you can imagine that some people have lots of genetic variants, each of which slowly nudges them towards the threshold, while other people have a single gene that greatly increases their risk. Now, one of the things you can see from that is that you can imagine the number of steps is along the bottom, so that by the time you reach the onset, you've reached your 6th step. So that's how you'd fit the multi-step model into this gene time environment model I was showing you earlier. But the other thing you can deduce from this is that if you're going to look for environmental risk factors, rather than just looking in the general population, you might want to look at people who have a genetic basis, a Mendelian genetic basis for their motor neurone disease, because they've already accounted for many of the steps. Then you've only got 1 or 2 steps to look for. It's likely to be an easier target than people with only a very small genetic contribution, or perhaps none, where you're looking for something that's going to affect all 6 steps. So maybe we should look for environmental risk factors in people with genetic ALS. So I'm going to stop there and just say, in conclusion, genetic and environmental risk factors contribute to ALS risk, possibly through a multi-step model. And the themes that seem to be emerging as important in causing ALS are DNA and transcriptome damage, cholesterol metabolism, and neuroinflammation. And I'd like to thank all my sponsors and funders, and also my team who've done most of the work that you can see. Thank you very much.
Yeah. Thank you so much, Ammar. That was absolutely fascinating and amazing just to see all of those different points coming together at the end. We've got 5-10 minutes for questions. I'm sure there's lots of questions. Should we start with Lizanne? Actually, I'll get another. Yeah. Thank you.
Thank you for this beautiful presentation and overview. So when you were talking about heritability, one of the pieces of data that you showed us is that 44 identical twins showed disease discordance. So do you believe from the data that that discordance is caused by genetic mosaicism, epigenetics, or environmental causes, or potentially a mixture of, multiple things?
So it could be any of those things.
Yeah.
A bit of data I didn't show you is looking at somatic variation. So just for the lay members of the audience, somatic variation is where genetic changes happen after you're conceived, and as those cells divide to become you you can get genetic changes, you can get errors in the genetic duplication that are then transmitted to all the daughter cells as you grow. So you may have bits of your brain that have a genetic variant, but other bits of your brain don't, for example. So there's some work from Ricky Patani's lab that shows that somatic mutation, actually, that there's a big burden of somatic mutation that's greater in people with motor neuron disease than people without. So that may well be accounting for it, because of course that would differ potentially between identical twins. Environmental risk and therefore epigenetics may also differ, especially if one smokes or not, that kind of thing. But a lot of those environmental risks are actually genetically, you're predisposed to them. So for example, there's a very strong genetic contribution as to whether you choose to smoke or not. And whether nicotine affects your acetylcholine receptors a lot or not. So you'd expect twins to be concordant for those as well, and therefore for those environmental factors. So I think somatic probably is one of the big explanations.
Thank you.
I think if we had a question, Luke was.
Thank you.
There's Katie in the front.
Hi, I had a question regarding that really interesting mouse that you mentioned with the retrovirus. Do you know how well characterized that mouse is, and does it exhibit any hallmark pathologies of ALS, like the neuronal hyperexcitability or TDP43 proteinopathy or anything like that?
So it's not very well characterized to my knowledge. It's a lower motor neuron model, so it doesn't have a lot of the features of ALS, and I don't know what the pathology is like. I can find you the papers that refer to it though.
Yeah, that's awesome. Thank you.
I just had a question down.
Hi. Yeah, interesting that the shorter telomeres are protective. Have you got any ideas about what the mechanism might be there?
No, it's pretty fine, so I don't. Telomeres are repeat sequences, so it may be something to do with the shape of the repeat sequence. I don't know. It's difficult.
Sophia.
Hi, Ammar. Great talk. Thank you. I'm very interested in another mechanism question, I guess, that you mentioned about the HERV-K virus, and it just selectively kills motor neurons. I think that mechanism behind that could be like the holy grail question: why does it kill motor neurons specifically? Is there a mechanism known, and does it kill upper motor neurons specifically or lower motor neurons?
So, I mean, it's not, to be clear, it's not the virus that kills the motor neurons. The motor neurons die if they have the virus. Presumably, it's the immune system that activates in some way and targets the motor neurons specifically when they're expressing it, but ignores other cells that are expressing it. So it's probably to do with the way the motor neurons are expressing the endogenous retrovirus, just because they have a different transcriptome, so they'll be expressing things differently. That's the only mechanism I can think of. But Avi Nath would be somebody to ask that question to. So I'll certainly look into that for you.
Just a quick comment on this question. The comment is that I was involved with NewMeds, the biggest psychiatry consortium ever, and we looked at all the genetic variants in schizophrenia for SNPs or copy number variants with the decode Icelandic population and multiple farmers. And the basic thing at the end was that any genetic variant which could affect neuronal plasticity with the gene-dependent effect would increase the risk of schizophrenia. So schizophrenia was then very similar, being a disease which has multiple molecular mechanisms and for two-step inflammation aspects at second trimester of pregnancy and then later on at adolescence, which then changed the critical brain circuits, which were particularly affected by. So working on the brain circuits was actually a downstream way in which the conclusion was that you should work out its schizophrenia, because otherwise there are so many mechanisms you fell into it. So that was actually quite similar to what you were saying in a different way. And the second question, the question was that one thing you didn't mention was motor neuron senescence. Viral senescence is something that's very well known. And I was wondering whether motor neuron senescence, with the Chinese showed CHIT1 to be critical factor for causing motor neuron senescence, could be associated with motor neuron disease.
Yes, I think senescence definitely would be important. That's more in the cell biology area where I'm not expert, but Kevin Tolbert, for example, in the audience would definitely be able to help with that. But I'd just like to reference your schizophrenia comment. we've done similar genetic studies and shown that there's about a 14% overlap in the genetic risk of schizophrenia and the genetic risk of ALS. So there may be some common mechanisms actually between the two. So the kind of common mechanism of neuroplasticity. Thank you.
Question here. I just wanted to follow up on your answer to Lizanne's question earlier. So I'm not, and you may not be familiar, that's fine, but with Huntington's disease, there's a lot of interest recently for somatic expansion of the trinucleotide. And I was interested to sort of see the connection here with some of these other expansions. And I'm wondering whether anyone's looking into that, or if you know of anyone who's looking into the role of the expansion of the repeats as a risk factor, or potentially could explain some of these the penetrance issues that you alluded to.
Yeah, so somatic expansion is very important as a potential mechanism. The commonest expansion we see is in C9orf72. And the problem we've got is it's very difficult to assay because it's huge. It's not a case of, so most people have between 2 and 20 repeats. Most people have just 2 repeats. People with motor neuron disease may have hundreds of thousands of repeats. And so you can't easily measure that because you can't use normal genetic techniques to assay it. So generally people use something like southern blotting, and then you need a lot of DNA, and it's difficult to get a lot of DNA. And especially when you're looking for mosaicism, you need lots of bits of brain to look at. So it's a difficult question to answer, but it's an important one.
Yeah, I mean, in the Huntington's, they see the DNA mismatch repair pathway coming up. I don't know whether there's enough data from the genome-wide association studies, for example, to drill down into the C9orf carriers and see whether those other pathways might be enriched there. But
yeah, I think that would definitely be something to look at. There's roughly 10% of the European population has C9 expansion. So that would be a large number of people and may well be enough to do that.
Any other burning questions? Yes, just behind you.
To bring it back to the telomere length, you said there's an additive effect if you have shortened telomeres than C9orf72.
Yeah.
Would you be able to expand on that a little bit?
I can't, because we've literally only just found it, and it's difficult to understand why. But there is an interaction between the two, so that if you have both, you've got greater than just the risk of one or the other.
So maybe 1 or 2 more questions. Yep. Is Adam or? Yep.
Thanks for the great talk. I was really interested in your comment that the genetic causes are finite. And my question is, I suppose, when we think about the latest sequencing that's going on in UK Biobank, when does it end in terms of 1 million people? You know, your GWAS was about 30,000. Yeah, I'm really interested in your comment on where should we be placing our funding from a genetic side of things.
That's a really important question. So you're right, because in theory, if we looked at 1 trillion people, would we even find things like variants in hemoglobin level would affect it? I think there is obviously a case of diminishing returns. At some point, we found enough genes to understand. You have to come down to why we're looking at the genetics, and it is the pathways in the end. That's why we want to know what we don't know yet. We found lots of genes, but we don't yet know exactly the mechanism through which any of them causes motor neuron disease. Even SOD1 we've known about since 1994, 93. So what's that, 30 years? And we still don't know why SOD1 variants cause motor neuron disease. Once we understand why, then it'll be easier, I think, to design treatments that affect that why, that affect that pathway. So we should, in my view, keep looking for genetic variants until we understand, at least for enough of them, the pathways. And we're getting close to that. You know, the cholesterol metabolism, the immune system, the inflammatory pathways. We are getting close. So I think fairly soon we'll be able to say, actually, we now know enough about genetics that we don't look anymore.
So I just got a quick follow-up on the cholesterol. Is there an association with statin use or a negative association in that case?
So people have looked at that with variable results. Some people show that statins actually protect you, and other studies show that it's an increased risk.
Maybe just 2 more questions.
There are still families with inherited ALS for which we don't know the genetic cause. Yeah. Why is that?
So about 20% of people who have a family history, you find no gene of the known genes. That's got two explanations. One is they have a gene that we haven't found yet, and maybe it's a difficult type of variation, so we don't assay it very well. Like the C9 expansion mutation, for example, you can't detect using traditional means very easily. But the other explanation is that it's coincidence. 1 in 300 people will develop ALS. So you would expect at least sometimes 2 people in the same family will develop ALS by chance, not necessarily because they have a genetic predisposition to it in that family.
Hello, lovely talk. Just perfectly pitched for everyone in the room. I wanted to kind of follow up from the question about when do we stop sequencing? Because if your argument is that we're looking at variants that cause people to develop disease, but then, you know, once the bonfire is started, then actually it's the modifiers of disease that are more important. I mean, do you not think that tofersen shows us that targeting the causes of disease can be curative or at least pause disease, or should our focus is really being looking at those modifiers more like UNC13A that are determining how disease looks after it's initiated?
So I completely take your point. If we have a technique to target directly the cause of it, then we can treat it. But let's say we could treat every single genetic variant that's responsible for ALS in the same ways we can use tofersen against SOD1 variants. You'd still only be able to stop the disease in 20% of people. There'd still be 80% of people where you can't do that. If you then look at the genetic variants like UNC13A that predispose you but don't necessarily cause disease, or that change the rate of progression, even that's only going to contribute another few percent. I mean, UNC13A variants, for example, are seen in about 5 to 8% of people, depending on the population you're looking at. So even if you did all of that, you still not treat everybody. So I think we do have to understand fully why, what is the pathway? And the other thing we never really look at is whether ALS is completely random. You know, maybe in some people there is no cause, in which case, what are you going to do?
God forbid.
And Gethin, did you have a question? Gethin?
Yes, so question around has anyone looked at the genetics of people who are really slow progressing, or sort of secondary part of that, in people who carry those quite pathological mutations? Have there been any evidence of sort of secondary mutations being protective?
So looking for protective variants is really important, and I think that would be a nice way of beginning to develop new treatments. People have looked at slower progressing people with slow progressing motor neuron disease. So for example, even many years ago, we did that, where we looked at about 50 people with disease lasting more than 8 years versus 50 people with disease lasting less than 6 months. It's difficult to answer those questions because the numbers of people in those two groups is small, and you do need tens of thousands to find proper genetic signals. So we did see some hints of things. Other people have done it as well, using regression models and things like that. We haven't yet come to a proper firm conclusion using those techniques. So I think it's an important thing to look for protective variants. The other way to look at it, which I think is what you were hinting, is that you could look at people who should have a bad prognosis but actually have slowly progressive disease. And I think, again, if we can get enough people like that, what we've built up in genetics is consortia. So that Project Mine, that's really helped us answer those questions because you can get enough people in. So we probably need to start thinking about, within those consortia, developing actual subgroups that will look at people within those groups.
Well, I think we've probably got a million more questions. So there'll be lots